Hi, my name is Isabel, and I will be starting off our presentation on the protozoan parasite called the Cryptosporidium. To start off, we're just going to go over our table of contents here. So kind of what we're looking forward towards this presentation. We have the introduction, which I'll start with. Then we have our social impact, which will be explained by Kyle and Iridiana. Then we have our current events, which is Chris and Jinghua. And then we have our future implications, which is Jixuan. So briefly in our introduction, we are talking about the Cryptosporidium protozoa and the disease that it causes. So on our next slide here, it is a type of protozoa that acts as a parasite. Not all species of the Cryptosporidium act as a parasite. However, the species that affects humans does act as a parasite. And as I mentioned earlier, the Cryptosporidium is commonly known as crypto. So if you hear us referring to it as crypto, it is the same protozoa. And it causes the cryptosporidiosis illness in humans. So on our next slide here, protozoa are microscopic one-celled organisms that can be free-living or parasitic in nature. This was the definition of protozoa by the CDC. Then our next slide here, just talking briefly about the disease, we have variability. So within the disease, there's a lot of variability within all protozoa and specifically the Cryptosporidium species. So these are single-celled organisms. They vary greatly on their species. Some species can live on their own and others are parasitic. And as we mentioned earlier, the specific species that affects humans is parasitic. However, all species that live within the humans, are the parasitic type. These cryptospecies cause the cryptosporidiosis disease. Then on to our diagnosis. So this disease is transmitted via fecal-oral route, so accidental ingestion of oocysts from the infected human. So the reproductive cells from the crypto, but transmitted from the infected humans. This could be from swallowing contaminated water or unpasteurized milk or cider and is often related to the frequent contact with either infected individuals or animals like cows or goats. And then our last point here are the symptoms. So the symptoms for this disease are primarily GI system focused, with the main ones being diarrhea, stomach cramps, diminished appetite, weight loss, and vomiting. Usually these symptoms are present for around 2 weeks, and it takes anywhere from 2 days to 2 weeks from initial contact to fully contract the disease and show these symptoms. And then on our next slide here, just briefly more on the Cryptosporidium. We'll talk more about that in our social impact, current events, and future implications.
Next, we'll discuss the social impacts of cryptosporidiosis. Cryptosporidiosis has exhibited serious health concerns in lower-income countries. When looking at the global crypto infection data, it becomes apparent that Africa bears a substantial burden. Out of 31 countries classified as the lowest income group globally, staggering 77% are located in Africa. This highlights the urgent need to address the health consequences of cryptosporidiosis in this region. A study conducted by Guinea-Bissau revealed a distressing association between Cryptosporidium and excess mortality in children under 12 months, with this increased mortality persisting into the second year of life. It is worth noting that the true extent of the impact has only recently been comprehensively measured. Global studies such as the Global Burden of Disease and the Global Attentric Multicenter Study have provided estimates of the mortality, morbidity, and disability-adjusted life years caused by cryptosporidiosis in different age groups and countries. These studies have highlighted the long-term consequences, including impaired growth, heightened susceptibility to other infections, reduced cognitive development, poor school performance, and an elevated risk of cardiovascular and metabolic diseases later in life. To put numbers into perspective, Dr. Ibrahim A. Khalil, in a research report on the Lancet Global Health, said cryptosporidium infection in children under 5 years was estimated to be associated with 44.8 billion diarrheal episodes and 48,300 deaths globally. Shockingly, Africa accounted for 75% of the diarrheal episodes and 88% of the deaths. Sub-Saharan Africa, particularly Nigeria and the Democratic Republic of Congo, bears the greatest burden, accounting for approximately 48% of the under 5 associated deaths. Furthermore, accounting for the downstream effects of growth shortfalls associated with cryptosporidiosis suggests that the actual burden of this parasite could be 2.5 times greater than previous estimates. Next, we will discuss cryptosporidial infection in livestock, focusing specifically on calves. Since crypto is primarily transmitted through feces, calves can quickly pick up the disease since they live in an enclosed environment. Direct contact between calves or indirect contact via contaminated objects or humans, as well as contaminated food or water sources, can also be transmitters of the disease. Managing cryptosporidiosis requires strict hygiene practices, especially during a cow's early growth. Calves should be born in a clean environment and adequate colostrum should be provided to them at an early age. It's crucial to separate calves and avoid direct contact between them for the first 2 weeks of life. Diuretic calves should be isolated from healthy ones during the course of diarrhea and for a few days after recovery. To prevent mechanical transmission, calf rearing houses should be regularly cleaned and dried between batches, while also controlling pests and rodents. While vaccinations have been attempted, they have not yet proven to be effective in preventing cryptosporidiosis. Controlling cryptosporidiosis in calves is challenging due to the nature of their habitats and the lack of a vaccine. Cryptosporidium infection in birds can manifest in various ways, depending on where the parasites choose to reside in the body. While some birds may not show any symptoms, others may exhibit signs related to digestive or respiratory systems. These signs can persist for several weeks and include diarrhea, weight loss, breathing difficulties, and even death. The two types of cryptosporidium in birds are respiratory cryptosporidiosis and intestinal cryptosporidiosis. Respiratory crypto affects the respiratory tract, leading to symptoms associated with breathing and sinuses, while intestinal crypto affects the digestive tract, causing digestive malfunction and malnourishment. Contaminated feces, water, or feed, exposure to infected soils, litters, or dust, and contact with contaminated human handlers or equipment can all contribute to transmission.
Cryptosporidiosis is a disease that is transmitted through contamination of food or water. The latter is an important piece because water is a vital part of everyone's existence, yet many countries are still facing the battle of access to clean, healthy water. Due to the way the disease is transmitted, the living conditions that a community lives in could greatly affect the risks and the risk factors of contracting cryptosporidiosis. There are cases of it in the United States, however, they're much more prevalent in developing countries. Kyle previously spoke of Africa as one of the continents that struggles with this. However, it is only one of many developing nations that are living under neglected conditions still. I want to talk about the socioeconomic reasons as to why some areas of the world suffer more than others. My first point is water quality, as the disease can be transmitted through contaminated water. A recent study was done involving several Southeast Asian countries such as Thailand, the Philippines, Malaysia, and Vietnam. They tested the water of these regions and found elevated levels of oocysts in the samples of the water taken from these areas. These levels indicate that there is an increased risk for zoonotic transmission of the disease in developing countries such as those in Southeast Asia. Treated water samples included any drinking water, tap water, and other sources of water that people from these regions rely on daily. The treated water contained no traces of oocysts, which means that they can be eliminated through this process. But this is an extensive cost that many developing nations don't have the means for. These 4 developing countries have a population of about 290.5 million people altogether, those of which who need to be on the forefront of policymakers' plans when taking into consideration public health in these developing areas. As I discussed before, poverty and poor living conditions are correlated with increased infection rates than those who live in wealthier developed countries. Looking at it on a smaller scale, right here in the United States, the same kind of inequality exists within our own communities, not just with cryptosporidiosis, but with other diseases and as previously witnessed with the COVID-19 pandemic as well. This study targeted the notion that the occurrence of this disease in the United States is independent of social inequalities and issues regarding marginalization. They found that demographics has a strong correlation with seropositivity, the rates being higher among Hispanic, Blacks, Mexican Americans, as well as other marginalized ethnic groups. These risk factors could be due to lack of knowledge on this disease and how it's contracted, and lack of access to healthy living conditions.
When looking at the recent current events with Cryptosporidium, it becomes clear that there have been numerous outbreaks. During the time period of 2009 to 2017, there were 444 Cryptosporidium outbreaks that led to 7,465 cases in over 40 U.S. states and Puerto Rico. Each year, the number of outbreaks had increased by more than 13%. In most cases, the disease was contracted by swallowing contaminated pool water, swallowing water from outdoor water playgrounds, contacting infected cattle, and contacting infected persons in childcare. In order to prevent this from happening, the CDC recommends not to swim or attend childcare if ill with diarrhea, and to wash hands after petting cattle. As of right now, Cryptosporidium is the second greatest cause of diarrhea and death of children after rotavirus. Molecular and biological studies indicate that it is more closely related to Gregorine parasites than to Coccidians. The finding of Gregorine-like stages allows Cryptosporidium to complete its life cycle without its host cells. Because of this, scientists are able to open new avenues in the investigation of pathogenesis, epidemiology, and the control of Cryptosporidium. However, there are no new vaccines or drug treatments to control this disease, since it is very difficult to test and work on in a lab setting. However, new drug sequences have been found that might be effective, but more money is needed in order to research and test these new drugs to make sure that they are completely effective.
50 cases of Cryptosporidium infection have been traced back to a farm on an island off the south coast of England. The UK Health Security Agency has been working with the local authorities and the farm to prevent further infections. The farm voluntarily halted the animal petting activities, which were identified as potential source of the outbreak. Dr. Anand Fernandez, a health protection consultant, states that there's no ongoing risk associated with the farm, and new cases directly attributed to the farm are not expected beyond May 10th. Secondly, risks associated with the farm visits. Visiting farms and touching animals can expose individuals to various infections, including E. coli and Cryptosporidium. The article emphasizes the importance of practicing good hand hygiene, such as thorough hand washing with soap and hot water after being near animals or areas frequented by animals, and therefore preparing or consuming food. Symptoms and transmission of Cryptosporidium. The Cryptosporidium is a microscopic parasite that causes cryptosporidiosis when ingested. It can be transmitted through contact with contaminated water, food, or exposure to infect animals or their feces. The main symptom is watery diarrhea, accompanied by stomach pain, nausea or vomiting, fever, and potential dehydration. Symptoms typically appear 2 to 10 days after infection and last for 1 to 2 weeks. APHA involvement in E. coli and Cryptosporidium incidents. The Animal and Plant Health Agency, APHA, reports its involvement in outbreaks during the first quarter of 2023. APHA assisted with a Shiga toxin-producing E. coli outbreak that had links to the same open farm involved in previous incidents. A suspect organism was cultured from a pig sample, which had an identical strain to human cases. APHA advised on measures to reduce public risk, including better supervision of animal contact and improved hand-washing facilities. APHA was also involved in an ongoing investigation of a cryptosporidiosis incident linked to a milk vending machine. Therapeutic avenue update on the Cryptosporidium. The availability of complete genome sequences of Cryptosporidium species has provided opportunities for developing novel drug candidates. Various approaches, including in silico analysis, comparative genomics, and structure-based drug design, have been utilized to identify potential therapeutic targets against Cryptosporidium. Several classes of inhibitors targeting key enzymes and protein involved in the parasite's metabolic pathways, calcium regulation, surface protein processing, and synthesis have shown promise as potential drug candidates. Drug repurposing and screening libraries have also identified compounds with activity against Cryptosporidium growth. These findings highlight the potential for developing effective treatments for Cryptosporidium infections.
So let's discuss the future implications and utilizations of this Cryptosporidium, especially for the question like how knowledge of this protozoa can be utilized and will impact the future in both human and animal side. First of all, let's discuss the improved diagnostics. Advancements in diagnostic techniques for Cryptosporidium can significantly impact the future. For example, molecular methods such as polymerase chain reaction PCR have enhanced the sensitivity and specificity of detection, allowing for earlier identifications of the parasite in clinical and environmental samples. And secondly, which is the public health interventions. Understanding the epidemiology and transmission patterns of Cryptosporidium is essential for implementing effective public health interventions. For instance, studies have shown that the importance of water treatment strategies such as filtration and disinfection in reducing waterborne Cryptosporidiasis outbreaks. Targeted surveillance and educational programs on hygiene practice can also contribute to prevention and control efforts. And thirdly, the vaccine development, research on Cryptosporidium's biology and immune responses has opened avenues for vaccine development. Several vaccine candidates have been tested in preclinical and clinical trials, aiming to provide protection against Cryptosporidiasis. Experimental vaccines utilizing various antigenetic targets and delivery systems has shown promising results in animal models, so it has already gained some outcomes and success in some areas. And also, let's discuss the one single health approach. Cryptosporidium affects both humans and animals. Emphasizing the need for one single approach is really important. Understanding the zoonotic transmission dynamics and implementing the integrated interventions can help prevent and control infections in both populations. Collaboration between human and veterinary health sectors is crucial for effective surveillance and control measures. So let's discussing the future implications of Cryptosporidium from the aspects of environment in drug development. So, Cryptosporidium is associated with environmental contaminations, particularly in water sources. Therefore, monitoring the presence and prevalence of Cryptosporidium in water bodies and agriculture settings can provide valuable information for assessing contaminations, risks, and implementing appropriate management practice. And also for the research and drug development, continued scientific research on Cryptosporidium can lead to the identifications of new drug targets and development of novel treatment options. Efforts are underway to explore potential antiparasitic drugs and evaluate their efficacy against Cryptosporidium infections. So, it's important to know that the specific future implications of Cryptosporidium will depend on ongoing research and advancement in the field. Finally, let's discuss some possible future expectations on Cryptosporidium. First of all, we should enhance surveillance systems for Cryptosporidium infections, which can aid in early detection and response to outbreaks, such as timely identifications of affected individuals, implementation of appropriate treatment measures, and the investigation of potential sources of infection are crucial for containing outbreaks. In the future, it may be possible to routinely transfect a Cryptosporidium and maintaining it in a long-term culturing platform to identify crucial functional genes, their roles, and their possible drug and vaccine targets. Apart from this, being able to maintain stable subtypes of parasites, consider interactions of the parasites and potential drugs to assess the relationships between genotype and pathogenicity is really important. It would be possible to develop effective treatments and vaccines against Cryptosporidius in the future. That's all our presentations. Thank you for listening and watching the slides. Thank you.